Welcome everyone to our information session about the 2026 10x Genomics Single Cell Fellowship, which this year is focused on FFPE samples. My name is Marta Makilov. I'm with Millennium Science, and I have many guests and co-hosts with me today, which you will meet shortly. But let's jump to the agenda for today. So I will start with just briefly highlighting what the actually fellowship is all about. And then Jeff McDermott, Science and Technology Advisor from 10x Genomics, will give you a practical introduction into 10x Genomics technology. Then Kat King from 10x, who is Field Application Scientist, will give some information about support from Field Application Scientist team and basically around the fact that the sample preparation is the key. And then the most important and the most interesting part is actually fellowship project snapshots from Shanna, Andri, and Liam. And that will be followed by a panel discussion about their experience with fellowship and question and answers. So please, during our presentations, feel free to put your questions into the chat or into the questions and answers space on Zoom, and we will be answering all of them at the end of this session. All right, so what the fellowship is all about. So Millennium Science Fellowship Program was created I think 4 or 5 years ago right now. And this is a program really centered around having a community of experienced researchers that can really get support they need to start with 10x Genomics, as sometimes it might be a bit daunting at the beginning, and we are here to almost physically hold your hand and walk you through the whole process and make sure you will succeed. So benefits of the being part of this program is mentoring session with 10x Genomics specialists for like really emphasizing the feasibility of your project and that we really want you to be successful. Also personal care of the fellowship program coordinator, which is actually myself, which means with any questions, if you lose email addresses or you need something specific, you don't know what to do, you can always email me or call me and I will find the answers for you. That comes also with discounted 10x Genomics consumables for Chromium, Visium, and Xenium, which Xenium is the new this year, so it's extremely exciting. We were not able to offer it in previous years, so this is the first year when you can actually experience Xenium through this program. If you're choosing to use Chromium or Visium, we can put demo instrument in your institute if you don't have access to the instrument. So that's not having instrumentation is no longer a problem. When you complete the program, you will receive the certificate and also speaking invitation at annual fellowship forum and other events like this fellowship launch when we have 3 previous fellows as our speakers. And basically you end up on my VIP list. So any specific discounts or offers or events, you will know first. So you will become really a 10x Genomics Millennium Science Fellows Network. And this is really a fantastic group of people that we frequently interact with. Also included is fellowship exclusive merchandise, which includes t-shirts for all the t-shirts lovers, but not only. So application. The application is pretty simple. It's not like, DECRA application, 30 pages and lots of time putting into. It's basically the title abstract, which we want basically to focus on novelty, feasibility, and impact of your research. This is the feasibility here is quite important because it really matches with our view. It's 10x technology is amazing, but we really want to match projects with technology and ensure the successful outcomes. We will also ask to attach one-page CV and a letter of support from your PI. If you are a young PI, you don't need a letter to support yourself. Just please briefly outline that you are the PI. We are especially supporting early career researchers, but also young PIs actually fit in that area. So conditions to entry: no previous personal experience with 10x Genomics technology, so you never used it before. You are graduate students, early to mid-career researchers, so we think up to 15 years post-PhD, but of course we are not super strict like some grant application, conditions. So if there are some exceptions, just let us know and we will definitely consider it. That's not the reason to reject a good project. So this year we are focusing on FFPE samples. So basically, the samples that you need to bring to the projects are fixed samples. If there's anything that you want to discuss, something else, please let us know. We might be able to help, but I can't guarantee that. Completion of individualized training program, which basically means that we will give you resources, whatever is needed for you to have a full comprehension of what you're going to be doing, to understand what you're doing, just to make sure that this is successful. This is very tailored and individualized, and it's not very stressful. And experiments to be carried out in Australia or New Zealand, because Millennium Science is distributor for Australia and New Zealand, and we can't really have this program in other countries. So what happens next? Please apply by 16th of March. Selected individuals will be contacted via email. Most likely it will be very early April. And I will offer onboarding meeting, which we will talk about your project and next steps. And then there will be multiple points of contact just to make sure that you will be successful. So as you noticed, this really focuses about your success, and we really want to help you with it, because not all those who wonder are lost. Some of those just didn't try 10x Genomics yet. With that, I will finish, and I would like Jeff to tell us about the overview of what 10x Genomics is all about. Jeff.
Thanks, Marta. I'll just share my screen. Okay, I'm going to assume everyone can see that okay. All right. Perfect. I just want to start off by saying thank you so much for coming along to the fellowship launch webinar today. I've personally been involved with this fellowship program for several years now, and it's been really inspiring to see researchers go from their very, very first experiments, where they needed lots of assistance with, say, generating the data and understanding the experiment, through to publishing that data and then moving on to using our tools more routinely in their research. And in most cases, actually coming back and becoming an advocate for the fellowship program themselves, such as the people who are speaking today. So if you do plan on doing a single cell or spatial experiment this year and you're using FFPE samples, then I highly encourage you to go ahead and apply for the fellowship. And I personally, really look forward to having detailed conversations about everybody's exciting projects. Oh, there we go. All right. So I'm going to spend about 20 minutes today describing how you can use our products to obtain single cell and spatial information from your FFPE samples. But before I do that, I want to take a step back and start with a really simple question, which is, why do we even want to obtain single cell and spatial information from those FFPE samples in the first place? And I'm not going to shock anybody with this statement here, but we all know biology is immensely complex. It's very easy to state that, but it's a very underrated fact. And you think of these myriad of molecules and cells and tissues all interacting with each other in all kinds of complex and dynamic ways. So to make any progress in unraveling the complexities of this biology, you need the right tools at your disposal. You need technologies that provide insights at that fundamental resolution of biology, which is the resolution of a single cell. To turn that statement, biology is immensely complex, into something a bit more tangible for today, let's take this human FFPE ovarian cancer sample as an example. This is a real-life tissue, and you can see I'm showing a H&E image of this FFPE section. And just like all tissues, this is a complex mixture of cell types and cell states. They're all communicating and influencing each other. And to best highlight that, I've got the small cartoon here of the tumor microenvironment, which shows a complex and dynamic interplay between all the different cells. And as a researcher, you might want to better understand what's happening in this FFPE tissue. For instance, what's driving that cancer, or why is that cancer, say, responding or not responding to any given therapy? So whilst these are high-level questions, they may sound somewhat simple. The underlying biology you need to interrogate to answer them is actually extremely complex. So let's take a look at how you would have traditionally examined an FFPE tissue section in the past using, say, bulk RNA sequencing. And here we're going to ask, what is the expression level of all the genes in the transcriptome in this sample? But of course, the sample is a complex mixture of various cell types. So when we take that bulk measurement, in essence, you're just averaging out that biology. You do get a measurement of all the genes in the transcriptome, but it represents an average signal across the entire tissue. And certainly bulk sequencing has and will continue to have its time and place, but it will never be able to fully answer deep biological questions when you're just averaging a signal across the entire sample. So now let's introduce single-cell RNA sequencing. Again, we're going to ask, what is the expression level of all the genes in the transcriptome? But now we have the power to address this question on a cell-by-cell basis. So you're measuring whole transcriptome gene expression from one cell at a time, but a scale of thousands to millions of single cells. And this yields, an explosion of data where you can now deeply profile individual cell types and cell states from a complex tissue like this. I'll move from that little illustrated plot to the actual single-cell UMAP plot from this exact same sample. And you can see by doing single-cell, we have a significant deeper understanding of the cell types present. And you can see them all annotated here. But the question is, can you ever really fully recapitulate the underlying biology of the sample by just knowing about all the cells in the sample? And I'm not saying there isn't analytical utility in single cell for tissues. In fact, I think that complete opposite has been proven to be true. There have been groundbreaking findings using single cell, and it's transformed our fundamental understanding across so many disciplines. But cells don't just act as a single unit. They're not operating in a vacuum, and instead they're interacting and influencing each other. And that's where spatial biology becomes so important. And to put it simply, you're connecting the what with the where. Now, not only are you measuring transcripts with single cell resolution, but you also retain all the spatial context to those cells. So if you like, we've taken this middle UMAP plot, and now we've placed every single one of those cells back into the context of the tissue. And from this, you can see which cells are interacting with each other and how they go on to form, spatial niches like this tumor microenvironment. So hopefully from this little example here, it's crystal clear as to how important it is to use tools that offer insights at that fundamental resolution of biology, which is the resolution of a single cell. If you're trying to derive deep biological insights, then bulk approaches just can't deliver on that. And these days you do need single cell or spatial measurements. You might be thinking, this is all well and good, Jeff, you're from 10x Genomics. Of course, you're going to be here today telling us that you need single cell and spatial technologies. But I would say the ultimate proof of what these tools can deliver is actually in the scientific literature, where researchers such as yourselves have showcased just how important these measurements are. And over the past decade, single cell and spatial, it really has revolutionized our understanding of biology across a broad spectrum of disciplines. And we track publications out there in the literature, and there have been over 10,000 of them in which they've featured insights derived from using 10x Genomics products. And in fact, to be honest, I challenge you these days to pick up a journal issue in almost any biological discipline and try and find at least one article that doesn't feature some form of single cell and spatial data. So this exact need for measuring molecules with single cell or spatial resolution, that's what underpins the direction we take when building our products at 10x Genomics. And as you can see, we've built 3 platforms that each allow you to look at single cell biology with a unique perspective. We have Chromium single cell, Xenium in situ, and Visium spatial transcriptomics. And for the rest of my slides today, I'm going to cover how you can extract the most value out of your FFPE samples by using these tools. But let's take a step back and ask ourselves a simple question. Of the platforms that I just introduced, which of them are going to be compatible with FFPE samples? After all, these single cell and spatial tools, they're very cutting edge. Is it even possible to use these 21st century tools to obtain high quality molecular data from samples that are traditionally notoriously poor in quality or might even be really old in age themselves? To answer that question, is an overwhelming yes. It is now possible to obtain high quality single cell and spatial data from FFPE samples using any of the 10x Genomics platforms. I've been in this space for over a decade now, and to be honest, if you had came to me in 2015 when I first started in single cell and said, "Can I get this kind of cutting edge single cell data from FFPE?" I would have said, that's just not possible. So let's take a quick look at how 10x Genomics has made it possible. Traditional single-cell and spatial approaches have used reverse transcriptomic-based biochemistries in order to capture transcriptomic information. That means you need an RT enzyme that makes a copy of the RNA molecule itself. But in FFPE tissues, the original RNA molecule, it's often fragmented or cross-linked, which makes this process very, very difficult. And it's been well established that it is difficult to obtain high-quality data from FFPE samples using a reverse transcription-based biochemistry due to those reasons. So that's why at 10x Genomics, we thought very hard about this problem and came up with a different approach to measuring RNA molecules. For FFPE tissues, we developed a probe-based biochemistry for RNA detection, which is what we have now implemented across Chromium, Xenium, and Visium. There are, as you can see here on the slides, nuances to how this works across all the platforms, but the fundamental premise remains the same, which is that probes bind to transcripts. And then for Chromium and Visium, these probes are turned into a sequencing library. So if you sequence probe A, you can infer that transcript A must have been present. For Xenium, these probes get turned into little balls of DNA, then we can then decode using microscopy. But either way, we're no longer doing any enzymatic reactions to capture the RNA molecules, which allows us to then generate high-quality RNA sequencing data from FFPE material. So now I'm just going to take a deeper dive into each of our platforms to show you the types of measurements that you can expect from FFPE samples, and we'll begin with Chromium single cell. So here's our Chromium single cell product portfolio. There's, of course, the instrument, which is up the top right, which through the fellowship you can access if you don't have one available. And then there are 3 classes of product families that can be run on that instrument. Of those product families, it's only the gene expression flex assay that is compatible with FFPE material, and that's because it implements this probe-based biochemistry that I just introduced. The core measurement of this gene expression flex assay is obtaining whole transcriptome gene expression from your FFPE samples. But do know, we've got probe sets that cover the entire protein coding transcriptome for human and mouse. However, it is also possible to spike in custom probes to RNA molecules that are not covered by those human and mouse probe sets from us. So examples I've seen implemented out there from people working in this space have been probes for things like long non-coding RNA, isoforms or structural variants, and viral and bacterial sequences. And here's the actual data from a number of FFPE samples that we've tested in-house. By no means an extensive list of tissues. Instead, the intention here is to represent many common cancer types, including pancreas, lung, breast, prostate, brain, liver, colorectal, and ovarian. So hopefully you can spot sample types here that either you work with or it's at least something that's analogous to some samples that you're already working with. So how do you actually go from your FFPE block to generating single-cell data? Well, at 10x Genomics, we've got a demonstrated protocol which steps you through the entire process. There are there are several different methods within this demonstrated protocol to choose from, and they include automated and manual versions of these approaches. And the demonstrated protocol also has a bunch of useful information about how to accurately count cells, along with various other things. So this protocol is going to be the complete guide you need to go from your FFPE block to suspension of single cells. We've tested this demonstrated protocol on a wide variety of FFPE tissues. In the table here, you can also find this will be in our demonstrated protocol, and we include detailed information on the size, section thickness, and cell yields obtained from the tissues we've tested. So I encourage you to access this protocol, not only to review the sample dissociation workflow and the different options you can take through that, but also to you get an estimate of how much tissue that you're going to need as input, as well as what cell numbers to expect as an output from the demonstrated protocol. And before we switch across to our spatial platforms, I just want to mention one final thing about the Flex assay, and that's that we quite literally just launched a new version of this assay, which we call Flex V2 or Flex Apex in our marketing literature. And this comes with some massive improvements to scaling up both cell and sample numbers, all while scaling down costs. So now in certain product configurations, you can obtain up to 1 million single cells from just one reaction. And on top of that, you can pull anywhere from just 2 samples to right up to 384 samples into just one individual reaction. As you can imagine, these advancements in sample multiplexing also come with the benefits of significantly driving down sample costs, as sort of I've tried to highlight in this plot here. So overall, it's a really great time to be thinking about running single cell on your FFPE samples, as with this new kit, you can now process more samples than ever, obtain even more cells than ever, and do all of that at a cheaper price than ever. So moving on from single cell, I just want to cover what spatial measurements are possible from FFPE samples. And before we dive into the world of spatial biology, I want to address a common question that I always get asked, which is, why does 10x have two spatial platforms and what's different about them, and ultimately what's going to be the right tool for my research? So we'll discuss each platform in detail, but at a high level, Visium HD is for measuring whole transcriptome gene expression, and it's a sequencing-based technology. And that means molecules are extracted out of the tissue, they're sequenced, and then they're put back into the context of the tissue by mapping them back to a two-dimensional grid. And you'll see that process in a moment, but we really see this as sort of an unbiased discovery tool. And then we have the Xenium platform, very, very different. It's a microscopy-based tool rather than a sequencing-based tool, so molecules are directly detected within the tissue itself and done so with subcellular resolution. With Xenium, we also estimate cell boundaries, so the platform generates true single-cell data rather than mapping molecules back to a two-dimensional grid. However, with Xenium, this is a targeted technology, so that means you need to select which genes to detect ahead of your experiment. So we see most people either running single cell or Visium HD for more of an unbiased look at the samples. And then we see people transitioning to Xenium to have sort of a subset of the transcriptome that they want to examine. But if you're unsure about what tools are best for addressing your research question, then of course, part of the fellowship process is going to be a meeting to discuss, what spatial platform would be best suited to addressing your biological question. So now you're familiar with those two platforms, I just want to briefly dive into each one. We're beginning with Visium HD. We'll start with an overview of what the measurement of this technology offers, and we're going to use this FFPE mouse brain section here as an example. So you still get a H&E or immunofluorescent image of your tissue that allows you to still do all the morphological analyses of your tissue, but you're also capturing the underlying molecular information in the form of whole transcriptome gene expression. For single-cell sequencing, these kind of plots, each plot represents whole transcriptome gene expression from a single cell. But for Visium HD, each point on this plot represents whole transcriptome gene expression from a position on a two-dimensional grid. And here's what happens when you take that signal and arrange it back to the two-dimensional grid. You're now able to recapitulate the underlying tissue architecture from just the molecular data alone. And you can clearly see now we have spatially resolved gene expression data. A quick snapshot of Visium HD data. I'm showing you results from a number of tissue types that we tested in-house during our product development. Just like the single cell example, this is not an exhaustive list of tissues that we've run, but instead it's intended to give you an idea about what to expect from some common sample types. So hopefully you can spot your tissue on this list somewhere, or at least something analogous to it. And a lot of people ask me, and it's on here as well, you can see there are things like tissue microarrays as well that are compatible with Visium. So I just want to cover an important question that everyone immediately has for me once learning about Visium HD, and that is, what's the actual resolution of the technology? So each Visium HD slide contains two capture areas. Each is 6.5 by 6.5 millimeters in size. And impressively, each capture area is a continuous lawn of oligos that's broken down into roughly 10.5 million 2 by 2 micron squares. So from each one of those squares, you're capturing whole transcriptome gene expression from the tissue above. But a 2 micron square, it's really a subcellular signal. So how do we take all those 2 micron squares and combine them together in a way that represents single cells? So in the left center image, you can do something just arbitrary binning, and that is taking 16 of these 2 by 2 micron squares and combining them into a larger 8 by 8 micron square. Whilst this is single cell scale and you can treat it like single cell data, the reality is this really isn't single cell data. So that's why we've also released a new feature in our analysis pipeline that uses machine learning to find the position of nuclei based on the H&E image that you take of the tissue as part of the workflow anyway. And then 2 micron squares can be mapped back to these individual nuclei, or even then mapped back to approximations of single cells. And as a quick example of this, here's a colorectal cancer FFPE sample. We're zoomed into sort of this single cell level. And you're seeing that the data has initially been binned to these 8 micron squares, which is why it looks pixelated. So now we're asking, are there any CD4 positive T cells that are infiltrating into this tumor? And you can clearly see outside of the tumor region, we've got a cluster of them, but we don't see any within. Now we take the exact same data and we're just bidding it differently, reprocessing it through the analysis pipeline. You can see that we now have a closer approximation of single cells. And using this, we can now identify CD4 T cells not only outside of the tumor region where we found them before, but we can also find some that have infiltrated the tumor as well. So it's just the same data in two different ways. All right, the last couple of minutes, I'm going to cover our second of our spatial platforms, which we call Xenium. Just like the Visium example, I want to start by showcasing the type of measurement this platform generates. We'll step through the different layers of data using this whole mouse pup as an example. And to start with, you can see that you can also generate a HD or immunofluorescent image of your tissue. Again, it allows you to do all the standard morphological analyses that you would typically do out of a tissue section. And then with Xenium, now we're going to layer on the precise molecular information. Since the technology is capable of subcellular measurements rather than looking at the whole sample, I'm going to zoom in and focus on the kidney. And now we're at the kidney, I'm going to zoom in and focus even more at the single cell level. So here we are with our histology, but this is where our histology journey is going to end and our Xenium journey is going to get started. So Xenium is essentially a highly sophisticated molecular microscope. It works by taking multiple images of the sample. And the first set of images collected are of different stains used to help identify cell boundaries. So during the sample preparation, a stain is added to the tissue. The stain contains antibodies for cell membranes, antibodies for interior proteins, a cytoplasmic RNA stain, and a nuclei stain. And then what you see here on the slide is a composite image of all those stains. And the whole purpose of generating these really cool-looking pictures is to find and segment cells. And you can see this is very different from Visium HD. Here on Xenium, we have a much more accurate representation of cell boundaries. Of course, we're not just using Xenium to find and segment cells. The core measurement of the technology is locating individual mRNA molecules directly within the tissue itself and doing so with subcellular resolution. So that's what you see here. All the dots on the screen represent an individual molecule that's been detected. And then we can stack all this information together. We know where transcripts are, and we have an estimation of cell boundaries. So any transcript that falls within the same cell boundary is now a single cell. And the instrument uses standard methods of single cell analysis to perform clustering of similar cell types and cell states. So back to a HD image of the tissue, you can overlay the precise molecular information and completely transform that standard histology type. You can see which cells are close to each other, how they interact and influence each other, and how they evolve in the system that you're studying. I'll note there are many different ways you can visualize your in situ data, and this is just one example. And also remember, we were really zoomed into where that little red box is, but if we zoom back out to the entire tissue section, you can see that this subcellular signal was generated across about 1.4 million cells in this sample, which represents about 177 million transcripts. I'm at time, so I'm just going to briefly mention these two things. The first is how much tissue area you've got on a Xenium slide. So each Xenium slide has 10.5 by 22.5 millimeter area in which you can place tissue, and each Xenium run runs 2 slides at a time. So here's some cartoons of tissue, but here's some real-world examples that I quite often find as they're posted on social media and add to this slide deck of how people are utilizing this capture area on Xenium. But the take-home message is you get far more tissue than Visium HD. And then the final point to cover is how do you go about choosing the genes you want to detect on Xenium? And there are several pathways that you can take when doing that. We have our Xenium V1 chemistry, which is up to 480 genes. Or we have our Xenium Prime chemistry, which is up to 5,000 genes. So you can see at 5,000 genes, it's a quarter of a transcriptome. It does start to blur the lines a little bit between Xenium and Visium being used for a discovery tool. So with that, just again, I want to say thank you so much for coming to the webinar today. The key take-home message is no matter what platform we've got at 10x Genomics, whether that's Chromium, Xenium, or Visium, it's all compatible with FFPE tissue. So I'll leave you on this slide here, which I think is a good segue into Kat's section, which is really sample prep. And how do you know you're going to get good results from your FFPE section? And it really starts with good sample selection. But Kat will explain a bit more about this in a moment.
Thanks, Jeff. Just a reminder, everyone, I see questions coming in the chat and Q&As. Feel free to pop these questions. We will answer them live at the end of the session. There are no stupid questions, and we will answer all of them. If we go a bit over time, the session is recorded.
Auditorium from 11 a.m.
The session is recorded, and we might go a bit over time. If someone needs to drop off, then you will be able to watch the recording and the answers to these questions. Kat, please tell us about sample prep.
I will. Hi, everyone, and nice to speak to you today, and great to see so many people interested in joining the fellowship this year. My name is Kat. I look after the field applications team here in Australia and New Zealand. And as Marta and Jeff mentioned, I'm going to talk to you today a little bit about sample prep. I don't have a lot of time to delve into this in detail. I more just want to mention what's out there and what you'll be supported with, as being part of the fellowship. So we know that sample preparation is, it's a really key part of getting really nice data from your 10x assay. The work at 10x assays, all of them, we have a nice workflow, they're end to end, but the key input that comes in is really going to define what your data looks like at the end. So we have, lots of resources out there to help customers with this. So how do I know which samples are going to be successful with the assay? How do I QC my FFPE blocks? How do I section them appropriately for use with each of our individual assays? And we've got these resources for all of our platforms, for Visium, Xenium, for Flex. There's actually quite a lot out there. General QC considerations. This is very general because I'm talking to all assays right now, but we want to understand a little bit, like Jeff already mentioned, around the tissue processing. Do you know, when that sample was collected and put into that FFP block, what was the time delay from collection of the tissue to getting it fixed? Do we know the fixation conditions? This will all play into how high quality that sample is, how intact that RNA is in order to perform well with our assays. We do use something called a DB200 assessment. This is where we look at the RNA and we see how long are the fragments, how long are the intact fragments after they've been through this FFPE processing and this fixation. And we've got recommendations around what might work well with our assay and which ones may, a little bit too compromised. We also like to look at our section quality. We've got recommendations about how to, section our tissue, put it onto slides or get scrolls for Flex. But we want to know what's the tissue looking like? What's the morphology? Do we have any necrotic regions that are going to perhaps not perform as well? So all of these things are contained in this guidance we have online on our support website. But what I really want to kind of mention is that can be overwhelming, especially if you're new to our assays and that you don't have a lot of experience with this and you don't know what the QC is, and there is a lot of documentation out there. So I wanted to also introduce the support team and who we are. So James and myself are the field application scientist team that work across Australia and New Zealand. And our role, as part of this fellowship is that if you are, within the fellowship, you purchase your kit, we will be in touch. And our role will be to talk you through, hey, how do you assess your samples? Where's all these resources? What do I need to know that this sample can perform well? How do I section my tissue blocks so that it's correct in the correct format? It's, following all the correct guidance for a successful 10x assay. So that's what we will do, and we'll have those conversations with you. We'll also be there for any kind of workflow guidance along the way. We're kind of your go-to for, running the assay and getting your data out. And if anything comes up and you're not clear of, that's where you can reach out to us. And also you get your data. How do you look at that data? How do you QC it and know that, hey, it looks as it should, the metrics look good? How do I process it using the 10x software pipelines? That's us, and we'll be supporting you with that, and we can then give you guidance about how you might use other platforms out there to start exploring the data further as well. So yeah, that's it. I didn't have a long time today, but I just really wanted to say that is part of the fellowship and how we will support you.
Thanks, Kat. I have a feeling we will run over time, but now we have the most exciting part of today's talks. We have talks from our fellows. So first we'll go Shanna. So Shanna did her fellowship when she was at Peter Doherty, and she's now moving to WeHi. She did some Visium studies regarding prions in brain cancer. Shanna, please tell us all about it.
Thanks, Marta. I'll just share my screen. Cool. Can everyone see my screen okay?
Yep.
Okay, thanks. So before I start, I just wanted to thank the 10x Genomics team and Millennium Science team for the invitation to share my experience with the Visium spatial transcriptomics platform that I was able to use throughout the fellowship program. So I've recently completed my PhD at the Peter Doherty Institute, and I was awarded the fellowship during my PhD, which allowed me to harness the Visium transcriptomics technology in order to examine the effect of altered prion protein expression on the tumor microenvironment. And this was really to tackle the overarching aim of my PhD, which was to investigate the role of the prion protein in glioma. So I'll just start with a bit of background first on my project. So gliomas are the most common primary brain tumours, which are categorised into low and high grade based on histological and molecular features. So high grade brain tumours are associated with poor rates of survival and also increased tumour recurrence, which really reflects the need to investigate novel biomarkers or alternate therapeutic targets that can improve patient prognosis. So my project focused on the cellular prion protein, or PRPC, as a novel biomarker or potential therapeutic target in glioma, given evidence of increased expression in several cancer types, and also the correlation between this increased expression and increased tumor grade, as well as reduced patient survival. So this increase in PRPC expression might perturb or exacerbate the physiological functions that have been attributed to this protein, which have been linked to the hallmarks of cancer. So to examine the effect of altered PRPC expression on tumor initiation and progression, I used a Cre-inducible mouse model of glioma, which involved deletion of the PTEN tumor suppressor gene and activation of an oncogenic mutation in PIK3CA. So essentially this was like removing the brakes and then pushing the accelerator down on the PI3K pathway in this model, which is commonly mutated in glioma patients. So I used tamoxifen to activate these mutations, or a combination of these mutations, in mice with variable PRP expression, and then culled mice at 6 weeks post-tumor induction, in addition to age-matched controls. So Jeff really nicely went through kind of the technical aspects of Visium spatial transcriptomics. So I use this technology as a really nice unbiased approach to understand the effect of altered PRP expression on the tumor microenvironment by mapping gene expression back to the spatial context of the tissue, which, like Jeff explained, unlike bulk and single-cell RNA-seq, this anatomical information is typically lost. So this was especially important to my project because I was interested in correlating transcriptional changes in the tissue with distinct tumor morphology. So this is me on my first day of training. You can see me aligning my tissue into the capture area. And as you can see, I used the V2 technology, which captures gene expression in the context of these barcoded spots, so sort of at the resolution of cell neighborhoods. So I was really nicely able to become skilled and experienced in each kind of part of the workflow, from sample preparation through to library preparation, and then through to data processing and visualization, where I was able to learn coding to be able to analyze my data. So I believe, I think I was one of the first people at the Doherty to use this technology, and this was also really great because it allowed me to establish relationships with other research groups who are interested in spatial transcriptomics as well. So this is just one example of the spatial data sets that I was able to construct for the control and tamoxifen-treated mice with, as you can see here, with high-grade glioma tumours, indicated by the dotted lines. These kind of spatial plots here are the result of unsupervised clustering using the Surat package to identify spatial clusters within the tissue that are mapped directly onto the H&E images. So what's really nice is that these clusters mirrored brain structural anatomy, and I was then able to compare gene expression and functional program differences in tumours here in red, compared to the neuroanatomical region in control mice, where these tumours emerge here in red as well. So as you can see, these tumours are quite aggressive at 6 weeks. So to get a better idea of the contribution of PRPC to tumour initiation, I also looked at animals 2 weeks post-tumour induction. And I really also wanted to maximize the use of the capture area by embedding or dissecting down brains from different mice and then embedding it within the same tissue block to ensure I had 4 brains within the one capture area. So one of the key findings from my project highlighted that inflammatory and immune processes were differentially regulated according to PRNP genotype. And now just shifting gears, I also had the opportunity to translate my experience with the Visium technology into another project, which is investigating the neuropathology of SARS-CoV-2 infection. So this involved upskilling to the Visium high definition technology, which Jeff spoke about, which maps gene expression to a single cell scale, which we can visualize by these barcoded squares. This is unlike the V2 technology, which is more of a cell neighborhood resolution. So in this project, we're now investigating the effect of mild SARS-CoV-2 infection at day 7, 28, and day 90 compared to an uninfected control using differential gene expression analysis and also characterizing the immune cell landscape in the brain. So to finish up, I just wanted to leave you all with some key takeaways or benefits that I've gained through the fellowship program. So from a more technical standpoint, in terms of the Visium assay, it really is a valuable hypothesis generating tool that can help to guide future in vitro and in vivo studies, and the spatial data sets that I've generated as well through this fellowship sort of serve as a really nice long-standing resource for not only my lab, but also other labs to be able to mine once this data is published. So from a more personal development perspective, it's really allowed me to gain really great technical skills, not only being able to run the Visium workflow, but also troubleshooting and learning how to code to be able to analyze the data. And this has also led to opportunities for collaboration, where I've been able to apply my new technical expertise into other projects, and it does look really great on your CV as well. So I'd just like to finish up with my acknowledgements. And again, thank you to the 10x and Millennium Science team for all your support over the years. Thank you.
Thank you, Shanna. That was really wonderful and great overview of your project. I will just now move to Andri, and if anyone has questions to Shanna, you can also put these questions in the chat. So Andri is at QIMR. He did his fellowship using the FLEX assay, and he was working on brain organoids infected with viruses. Andri, tell us about this.
Yes, let me share my screen. Sorry, I'm having a bit of technical issues here.
It's okay. Maybe then if Liam is ready, because I saw Liam in the audience, he can jump first and then Andrew will go after?
Yeah,
sounds good.
Yep.
No worries.
All right, so we are still moving to Liam, but we'll go back to the viruses in organoids. Liam, I think you'll be talking about Multiome, and Liam is based in Monash. And as you can see on his front slide, he used the Multiome kit, which allows to test really gene expression and chromatin accessibility in single nuclei.
That's right.
Liam, tell us.
So yes,
we see your the view is like, what's called this type of view?
Oh yeah,
right.
Right.
The other type of view. Yeah, that one. That's good.
This is good?
Yeah.
Okay. Sweet. So I had the opportunity in 2023 to use the Multiome kit. And really at the time, and still, we were really interested in trying to define a signature for long COVID. So long COVID, or PASC, as it's often referred to, refers to a syndrome that is defined by persistent symptoms following a COVID-19 infection. Now, it currently has a huge burden of disease and cost. So about 5% of individuals who catch SARS-CoV-2 end up developing long COVID. And currently, there's no validated diagnostic or therapeutic approach to specifically deal with this condition. So past diagnosis currently relies largely on self-diagnosis, self-reporting, and therapy is still quite a while away, despite up to 400 million people now being affected by it. So we decided to use this novel sequencing approach to perform high-resolution analysis on the memory B cells that are formed and persist following a COVID-19 infection, in order to try and define a PASC signature and open up new avenues towards diagnosis and therapy. So in order to do this, we isolated PBMCs, or peripheral blood mononuclear cells, from individuals who had long COVID and individuals who recovered from COVID-19 without issue. Because of the Basically, because of how sensitive this approach is, we needed to control for a number of factors. And because there's a huge batch effect that can be associated with it if you don't control effectively enough, the approach that we took was matching individuals by time post-COVID-19, by age, ensuring that all of our PASC individuals had at least some overlap of symptoms, in this case, shortness of breath. They were all vaccinated and sex-matched. And what we actually did was, within our matched pairs, we actually pooled the cells together into the same tube and performed sequencing on these combined samples. Now, because of the sensitivity, we can actually demultiplex or separate our individuals based on SNPs and then actually re-identify them using their HLA information. So by running them through this pipeline, we were able to extract both RNA sequencing and ATAC sequencing information from the same cells and then separate them based on healthy or long COVID. Now, what jumped out to us immediately was the expansion of specific subsets of memory B cells. So those that were expressing markers associated with interferon experience, as well as atypical chronic B cells that are typically enriched in contexts of chronic disease. Now, when we actually looked at the frequency of B cells expressing genes related to certain pathways, we saw a statistically significant increase in those B cells that are expressing genes related to interferon pathways, or that come up following interferon exposure, highlighting that we actually do have an enrichment of atypical B cells that are that have been subject to interferon experience. Now, finally, one of the most exciting things that we were able to perform with this analysis was eRegulon analysis. Now, because we have RNA and ATAC information from the same cells, we're able to integrate the transcription factor expression using the RNA sequencing information with the accessibility of those transcription factors motifs, looking within our ATAC sequencing dataset, and finally look at the expression of genes that are downstream of those transcription factors. And by combining that information into a single activity score, we were able to identify and compare the contribution of different transcription factors to both healthy and long COVID B cell states. So without further ado, I'd like to thank everyone involved and say that this really was a fantastic opportunity that you should all consider. Thank you.
Thanks, Liam. That was really cool. Andri, how are your technical skills are developing? All right, Andri,
you got it working.
Organoids, viruses. Here we go.
So in our lab, we are studying human neurotropic viruses. And the project that I will be talking about is modeling West Nile virus neuroinfection in human brain organoids and understanding the mechanisms of West Nile neurovirulence using single-cell sequencing. So if you are not familiar with West Nile virus, it's a mosquito-transmitted positive strand RNA virus that can infect humans and horses as the dead-end hosts, and it can cause severe viral encephalitis that can result in fatal outcomes. And currently, there is no vaccine or specific treatment available for West Nile. And the mechanisms of West Nile encephalitis are poorly understood, and the information coming primarily from mouse experiments, which are generally poor hosts for flaviviruses like West Nile, and provide limited information that can be translated into understanding of disease pathogenesis in human. So at the time, we established brain organoid model of viral infection that we successfully used to study Zika virus. And we decided that we should use West Nile virus infected brain organoids and analyze them by single cell sequencing to understand which cell types become susceptible to virus and which responses they develop. However, we were facing several issues. First of all, at the time, most single-cell sequencing kits were based on poly-A capture. However, flaviviruses such as West Nile virus, they lack poly-A tail, and therefore viral RNA cannot be effectively captured in these libraries, which will make it very difficult to tell infected cells apart from uninfected. In addition, West Nile virus, depending on the strain, it's risk group 2 or 3 pathogen, and infected cells cannot be removed from the containment facility to take them to the cell sorting or library preparation instrument. And this was when I was starting this project. I was awarded an 10x Fellowship, and this is when, from chatting to the 10x expert, I learned that 10x has recently released a new platform which solves all our issues, and that platform is the 10x Flex platform, which is probe-based. First of all, it is possible to design the probes that will capture viral RNAs, and that this will enable detection of virus-infected cells. In addition, it works on formalin-fixed samples, which solves all our biosafety and biosecurity issues. So, working together with 10x support team, we designed the probes that bind to the viral genome, and we ran the pilot experiment, which showed that virus-infected cells can be successfully detected using this platform. And we then performed single-cell sequencing of West Nile-infected organoids. We found that organoids contain a lot of different cell types, including astrocytes, neuroprogenitors, and different types of neurons. So they have very nice cellular diversity for us to study the viral cell tropism. And when we looked at the presence of viral RNA in different cell types, we found that most of the infected cells were primarily immature neurons, and some infection was occurring in astrocytes. We then looked at the expression of interferon beta, which is the main antiviral cytokine that drives the antiviral response to infection with positive strand RNA viruses. And we found that that small group of infected astrocytes, they were the only cells secreting interferon beta. And also we noticed that only a small percentage of infected astrocytes were producing interferons, which shows for the potent ability of the virus to inhibit the interferon response. And interferon works by binding to the interferon receptors in the susceptible cell types and triggering expression of antiviral genes, collectively known as interferon-stimulated genes or ISGs. And then we looked at expression of interferon-stimulated genes in different cell types, and surprisingly, we found that strong upregulation of these genes in infected and uninfected astrocytes and progenitor cells, but not really immature neurons, which shows that neurons, they not only they can't respond to the viral infection itself, they also don't respond to the antiviral cytokines. and to ask to understand why, we looked at the expression of so-called pattern recognition receptors. Pattern recognition receptors are the proteins that can recognize molecular patterns associated with the virus, such as viral RNA, and trigger the signaling cascade that ultimately ends up in upregulation of the interferon genes. And for detection of West Nile, the main pattern recognition receptors that detected are RIG-I and MDA5, encoded by IFE1 genes, and also in mouse, it was shown that it can be recognized by TLR3. Surprisingly, in human samples, in human tissue, we found no TLR3 expression at all, while RIG-I and MDA5, they were expressed in astrocytes and different glial cells and progenitor cells, but not in mature neurons. And also mature neurons lack expression of any other pattern recognition receptors that can detect a viral RNA. And this explains why neurons don't respond to viral infection by production of interferon. We then looked at the expression of the genes that regulate downstream signaling from the interferon, and we found that unlike astrocytes and progenitor cells, neurons lack expression of the kinase TYK2, which is the kinase required for transduction of the signal from interferon receptors to the transcription factors STAT1 and 2 that regulate expression of these antiviral interferon-stimulated genes. And this explains why neurons cannot respond to interferon that is produced by infected astrocytes. And at the conclusion, we showed that in the human brain tissue, West Nile virus primarily targets immature neurons, while only a small number of astrocytes become infected. And astrocytes serve as the main source of interferons that drive antiviral response in other astrocytes and progenitor cells. And neurons don't produce interferons and poorly respond to them, which makes them susceptible to West Nile virus infection. And we found that neurons' inability to sense viral RNA was due to lack of expression of pattern recognition receptors, while their lack of response to interference was due to deficiency in TYK2 kinase expression. And overall, it paints a picture in which astrocytes act as the major protectors of the brain from the infection with West Nile virus. Thank you.
Thank you, Andriy. Thank you. That was really interesting, your project. And due to, well, we are running a bit out of time, and our fellows already mentioned some recommendations to join the program. Let's jump into questions, because we have quite a lot of those. I will be reading them out loud, and I will answer them, or I will ask one of our panelists to answer. So let's start. How long it should be? Should it be part of our current project? If not, I guess fellowship. If not, where we are able to perform it, which lab? And finally, on which types of cells it's usable, animal or plant cells. Okay, so it can be part of your current project. You can run it, or it doesn't have to be, it's just planning something new. You can run it in a platform that is available in your institute or through a core facility, which we can connect you with. If you would like to have instrument placed in your lab, if it's Chromium on-site assist for Visium, that's also a possibility. And with the focus on FFPE, the majority of assays are easily adoptable for mouse or human samples. However, with the Flex Apex and with Xenium, it is absolutely easy to, maybe not super easy, but it is possible to run it on plant cells or run it on any other species. And there was a question also in the chat about ferrets, so like in ferrets. Just because Xenium, you can design custom panel, and with Flex Apex, you can design also only your probes. You don't need to rely on 10x Genomics probe set. but you would have to purchase these probes separately. So the next question is, what are you specifically looking for in the CV? So it's basically, what's your general experience, especially in terms of molecular biology? What are your skills? Do you know how to handle RNA? So it's just sort of basic assessment of where you are in your career and whether this fellowship will also benefit you. Is the length to the fellowship, in example, 2 years? So usually we assume the fellowship is designed to be completed within the 12 months. However, there is possibility to extend it a bit longer. Usually it can be longer than 2 years, but majority of fellows are able to complete it within the first 9 months to 12 months. And it's basically due to sort of optimizing experimental conditions and sample preparation and things like that. Which cost this fellowship covers? So the fellowship offers quite a lot of free support, which is usually not included on quotes, but it's a really extensive amount of time that the team puts an effort to. So it's really hard to quantify it. And the fellowship covers reagents. There is a 20% discount applied for the reagents. So you still need to have funding to use this fellowship. However, it's not only what matters within the fellowship is more the support, and we are making sure that you will be successful. So it's not really free kits. This way also it allows us to support much more projects than only one chosen winner. Are the kits included in the fellowship, or do we need your own funding? So I answered these questions already. In my project, I'm going to characterize a pan-cancer neoantigen-specific T cells and subsequently do single-cell RNA TCR sequencing for identification of proper TCRs to be used in engineering of TCR T cells. Can I apply for this fellowship? So you can apply, however, this specific question will not be able, and Kat, Jeff, James, please jump in. Well, you will not be able to assess this information from FFPE samples. Correct, Jeff?
That's correct. There have been some publications out there that have shown this kind of workflow before. So by all means, if you've only got FFPE samples, it's going to be tricky, but otherwise we can have a conversation around how to go about doing this.
Yeah, so you can submit the project and we can have this general discussion also to see how feasible it is with the samples that you have. And we will take it from there.
It's definitely something that's more feasible for projects based on fresh tissue or things along those lines because of the ability to do poly-A capture along those things. There are, as Jeff said, a couple methods out that people have done TCR, BCR profiling and things by spiking probes, but it's not as straightforward, especially with FFPE tissue and how well those sequences are preserved after processing. But definitely happy to chat to it, whether it's part of this fellowship or outside of it. It's something that a lot of people are looking at.
If I have previously used 10x Genomics products and equipment, but not specifically offered in this fellowship, can I apply? Well, that's a tricky question. So with our conditions, you're completely a new person to 10x Genomics, so that would disqualify you. But maybe you can drop us an email and we can make a case for in this specific situation. So definitely, we are happy to support your project, but whether it will fall into the fellowship, we will have to have sort of discussion within our fellowship sort of management team to see if we can accept that. Can spatial inference CNV be done on data generated from any of the platforms?
Yeah, there's a few custom scripts and projects looking at CNV type spatial interference analysis on some of the spatial platforms. So people have done it on Xenium, people have done it. I'm not sure about Visium so much, but it's definitely possible. Again, one of those things where it's not as straightforward, but it really depends on the type of analysis that you have in your team, especially from a bioinformatics side. But if it's there, we should be able to detect it. And they definitely have people looking at that kind of stuff, especially with Xenium data. I'm not sure as much about the single cell side. Maybe Jeff can jump in there.
Yeah, certainly I've seen it successfully done on the single cell side as well. Infer CNV is the third-party package. Again, it's not something that's written by 10x. We can't guarantee the outcome of CNV detection from our products, but I have seen people successfully implement it in the literature. Again, we can talk about it when it's time to have a project discussion.
Thanks both. How much is the price of the probes and the resources we need approximately? So I saw a few questions in regards of pricing. So the cost of reagents really depends on the project and which pathway, which assay you choose. So the cost for single cell for one sample can be as low as around $500, but it usually will be higher because that would require quite a lot of samples to run. However, what actually is a part of the fellowship, when we are going and having discussions, we are always proposing the most cost-effective solution. So there are many ways really to run your single-cell spatial transcriptomics, and our role is match the assay to your research question, but also to your budget. And Javad, who was mentioning the TCRs, he's not working with FFPE samples, so definitely your project is feasible, but because the fellowship is focused on FFPE samples, it kind of falls out of the scope of this program this year. What is considered an old FFPE sample, like more than 4 years? Have you seen differences in data obtained depending how fresh old samples are? And there was also another question related to that about can samples be 9-10 years old? So.
Yeah. It does depend on, we can always analyze what's there in tissue. And if the old samples have been fixed appropriately, stored appropriately, then we can do quality assessments on these blocks to have a look at the fragmentation level of these samples. In-house, we've run on some platforms from the spatial side samples that were 15 or so years old, and they've worked well, but commensurate to the quality of RNA that's there. I always say fresh is best, but we do have groups that run a lot of archived samples from patients that are years old. It always comes down to how much RNA is there in the tissue. Do we have, really bad fragmentation? A lot of other things. But the good thing about this is all of these samples, we have a similar QC methods we recommend, whether you're doing flexed or whether you're doing spatial on fixed tissue. We would look at things like the DV200 score, so the size of the fragments of RNA over 200 base pairs, and the threshold is relatively low, so we say 30% or above. The probes for a lot of our assays are 40 to 50 base pairs or so, sometimes smaller. So as long as that binding site's unfragmented, then you should get a detection event on paper. So that's a metric we would use. And if you have these older blocks, we would do some quality assessments just to see sort of what we're working with. If you can get fresh samples and you can fix them and put them into blocks, then that rules out a lot of the kind of variation you can get from time. And if you said to me, I've got a 2-year-old sample and a 10-year-old sample that were prepared the same way, fixed appropriately, etc., which one would you run? I would always say, well, which one has the most important biology in it? And if they have the same biology, I would say, okay, well, let's go fresh is best in that regard. So there's no real time limit that disqualifies them. You pull something that's been sitting at room temperature on top of a shelf for 40 years, probably not going to work as well as something that we put into a block 6 months ago. But definitely 3-4 years is relatively common for people, especially people doing things like TMAs and stuff that contain a wide multitude of sample qualities in them.
Yeah. Next question. Can we do both single-cell RNA-seq and ATAC-seq on the FFPE sections?
From the spatial side, no. ATAC-seq is not currently available as a spatial product in our platforms, no.
And single side too.
Yeah.
You can absolutely do single-cell ATAC, but just not with FFPE tissue.
Yep.
Okay, is there a limit to how many custom probes can be spiked in?
For the fixed base assays where we spike probes in, no particular limit. It is going into sort of something where we have a tech note on probe design and different considerations for Visium and the Flex assays, fixed assays. We don't validate these probes in advance, so it is the risk is taken on yourself. But yeah, we don't necessarily have a limit to the amount of probes you spike in. If you're spiking in a huge amount, then you potentially want to compensate in your hybridization buffers for that extra volume of probes. But given how many probes there are in the whole transcriptome base panels, not necessarily something you need to go in a crazy amount for. For Xenium, we have a limit with the different assays you do. So if you were doing a Xenium, you could do a fully custom up to 480 gene if you're doing 480 genes in the version 1 of the protocol, or you could do the 5,000 plex and spike in up to 100 genes on that. And you could also spike in up to 100 genes on any panel that's already pre-designed. So yeah, if you're doing full custom, 480 for Xenium, but if you were spiking in custom probes with one of our base panels, then the limit would be 100.
Thanks, James. Is the in-house Visium HD data of different tissues publicly available? I know that some Visium HD data is publicly available.
There's about 46 different samples that we have the full data set available, including all of our input FASTQs as well as the images we used. This is varying from, if you looked at the FFPE, this is usually human and mouse samples, but if you looked at the fresh frozen, we also have poly-A data generated in plants and rhesus monkeys and rats and a few other things on the website now as well. So yes, everything on our 10xgenomics.com/datasets, and then just filter up the top left by Visium or Visium HD. There's single cell data there as well, a ton of it. But yeah, I think we're up to 40 something data sets for Visium HD. So there's plenty to play around with. And especially if you want to get your analysis pipeline up and running before your samples are ready to run, you could have basically everything downstream already sorted out by using our data and downloading it as if it was your own, which will speed you up in the long term.
Is it still possible to apply for the fellowship program if also applying for a discount through other avenues like Research Accelerator program? So just the 2 discounts wouldn't be applied at the same time. That's for a start. So these programs are a bit different. Fellowship program is heavily focused on support, while the Research Accelerator is really program which is very short-living. So it will have a very quick deadline in this current accelerator programs, like end of the 20th of March or something like that. So if you are able to run or purchase the reagents before the deadline, you will probably get better pricing through the Research Accelerator program. However, if you require more support, you're not ready, then definitely fellowship program is something that you should consider, because it will lock your support and lock your pricing for you for a really extended period of time. What's the gene coverage using the FlexKit Visium and Visium HD? What's the rough possible cost that I answers? Do I need to have the samples ready right at the start of the fellowship? That I say you don't have to, but you need to have it within the period of time that the fellowship is designed for, so within the year. And we talk about it at early meetings. So what's the gene coverage, Flex and Visium?
It's whole transcriptome. So the lovely thing about this is the Flex and the Visium both use the same probe set. So somebody else asked about combining different methodologies like single cell and spatial and things. So for Xenium and Visium, they use the same probe sets, which is great. Usually whole transcriptome coverage, there's a few genes that are harder to design probes for and a few other things. So generally it's coding genes, usually sort of somewhere in the vicinity of 19 to 20,000. Doesn't necessarily mean that you would detect all of them in your tissue because of just biology, but it should be whole transcriptome coding. And they're generally designed to cover as many isoforms as possible. So we can, if you've got, a lot of more highly expressed isoforms than others in your tissue, generally they're designed in exonic regions that translate across multiple isoforms. So it is something to consider if you're wanting to target specific ones. That's where the custom spike in might be something to consider as well. And just to segue on that, someone asked about Chromium and Xenium combining the data. Yes, that's definitely some way that people will do it. Often we actually use the single cell data to help us when we design our custom probes for Xenium. We find there's a really good correlation between single cell data and Xenium data. So if you're generating whole transcriptome, Flex data, for example, and then from that you pull out a gene list that you want to design a custom panel for and investigate from a spatial side, then yeah, they work really, really well together. together. And similar between our Visium and our Flexen as well, find a really good correlation because of those shared probe sets. So definitely something that people commonly do when they want a bit of both worlds there.
Okay.
With the probe sets, they're available on our website. So you can actually go and have a look now at what the probes are targeting, across those panels. 18,000, I think, around 18,000 for human, 21,000 for mouse. They're available as a CSV file.
Yeah, and it's based on the MM39 and the latest annotation of the human transcriptome as well. So yeah, should be relatively up to date.
Okay, next question. We have still a few questions, and I'm devoted to we answer them all. With single-cell RNA for plants, do you recommend specific protoplast isolation protocols? So this is a bit sort of tricky questions as we are talking about FFPE samples, although I would assume that would be like really longer discussions, because with single-cell for plants, theoretically, with the fact that Flex, Apex can be purchased gem separately and design probe separately, it is still possible. However, that would be probably a bit difficult. But I think in general, for fresh samples for plants, do you have at 10x Genomics has protoplast isolation protocols?
We do. Oh, sorry. You go, Jeff.
No, okay.
We do have a protocol for protoplast isolation. Most people that I've worked with who work on plants, plants, there's so much variability out there. It depends what you're working with. There's protocols in publications that people often refer to. We've got limited guidance, on working with plants in terms of actually developed protocols that you can just follow. And the publications out there are probably your better resource. We always work with customers to, help them implement those protocols. The idea is to get nice, high quality samples to feed into the assay. That's the key. So you don't need to follow our protocol. It's more about how do you get that nice, high quality, clean sample. And yeah, we've got lots of experience kind of troubleshooting that and getting nice single cell samples to feed in. Yeah.
Okay. Can bioinformaticians apply for the fellowship if planning a project this year with no prior personal experience? Yes, of course you can. It's usually the question is it that's your project? Is it a project with someone who will be actually doing the manual work? Because some people do both analysis and manual work. So you might consider in this case, perhaps it is rare, but you can submit joint applications. So 2 names under 1 application. That would be fine. What kind of statistical power does the FFPE approach have? How many samples biological replicas should be run or can be run in the fellowship? Jeff loves this question.
I think I don't have a spare 45 minutes to go over it all in great detail. Let's save that for a project discussion. I think that's the best way to answer that question.
Yeah. Yeah, exactly. Because
it depends on your biology. What do you want to measure? What other assays you've already
and on the
budget. There are many factors to consider when thinking about biological replicates and power of the number of samples you want to run.
Okay, next question. Do you have an estimation of the scheduled timing from applicant selection to the experiment being carried out? Well, it depends really on your project and on you. Do you have samples? How much do you need to learn from the start to running the experiments? So it can be as soon as, within 1 month, or it can be longer. In terms of FFP quality, this is about the 9 and 10-year-old samples, blocks. Can we still apply if I haven't gotten a grant to carry out the project yet? Getting the fellowship would strengthen our applications to funding bodies, but we are not sure when we will be able to carry out the experiment. So we encourage, you need to have funding in this specific fellowship program to actually run it, but we will not reject good project if you are applying for funding. And one of really strength of this program is to really support you in terms of that you can put it in CV and it actually strengthens your application, because we can have these early discussions and you will get more knowledge about your essay, and we can help you really putting the application together in a more emphasis on feasibility. Okay, we have that question was answered. Okay. So what is considered previous experience with 10x Genomics? Basically, if you purchase reagents from 10x Genomics, you run the experiments yourself or through the core facility, that would be considered a personal experience. And next question, has anyone ever performed proteomics on the same tissue slide afterwards with success?
Yeah, definitely something that you would be looking at using the Xenium for that. With Visium and Flex, obviously, it's going to be destructive on the tissue to an extent, because we have to homogenize the samples, and for Visium, we have to do the transfer and the permeabilization of the tissue. But for Xenium, it's non-destructive. So, yeah, proteomics is very commonly run downstream. You can actually do proteomics inline if you're doing a Xenium version 1 assay. So if you're looking at the under sort of 500 gene mark, you can actually, we have a protein panel of up to 27 markers that you can use on the same samples. Again, non-destructive. And then there is also the option of doing things like, third-party platforms that do cyclic IF, as well as doing your own IF post-run. So basically what you're left with is tissue on a slide, and you can really kind of do whatever you want with. There's a couple considerations about things like antibody species. So our cell segmentation markers, for example, are raised in rabbits. So you'd want to avoid anti-rabbit, but all of the fluorescence should be stripped off the tissue. So you can really do whatever you want downstream. But a little bit harder with other platforms where the tissue is homogenized or destroyed for sure. In that case, there's always the option of doing serial sections.
How much is the price of the probes and resources we need? So there are some costs also related to third-party reagents, which the list will be provided to you. There need to be some specific reagents, and if you use any spiking probes or your own probes in certain cases, then 10x will not provide them. The other probes within the kits are part of the kits, so you will have to source this pricing separately. Does it make sense to combine Chromium and Xenium? Then James answered that question. It's really actually a cool way to do it. I assume we don't have to indicate which platform system we are going to use in the abstract, as this will be discussed with the team later. Yes, that's exactly the way, unless you really have already dived deeply into what the platforms can do and you have some specific preference, you can mention it, but that will be definitely discussed in the initial meeting. For people without funding who may need to try and acquire funds for the Visium Spatial HD brain slice, to do the spatial sequencing plus reagent, what is the estimated cost per sample with the discount? It could be possibility to relocate funds or gain some pilot funding from the university. Okay, so with that question, if you could drop me an email, I can send you the specific calculations. And because on our side of things, we can only sell reagents as the kits are sized in. And when it comes to cost of sequencing, especially with Visium HD, then it's additional part of the costing. So I can connect you or check in my previous sort of quotes that I have how core facilities could possibly cost you for this part. And we work closely with core facilities so we can do it.
And we can also advise you, I guess, on the recommended sequencing depth for you, depending on what platform you want. And then from there, you can have reach out to different sequencing providers to work out sort of what you'd need for that. So that's definitely something we talk about because, each sequencing provider has a different charge and different configuration. So we'd at least leave you with knowing how much sequencing you needed for your samples.
Yeah, and the last question, I had a project through AGRF where they used Flex and handled bioinformatics. Am I still eligible? But really, it sort of is within the I had experience with 10x because you sort of used through the AGRF, so the kids, so not really eligible for this program, but there are many other programs. So if you would like to specifically benefit from this fellowship or other sort of interesting programs that we are running, also feel free to just drop us a line and we can see what we can do for you. With that, I think we answered all of the questions. Thank you so much for the interest and also staying with us. Thank you for all our panelists and fellows for these great presentations. And I'm looking forward to seeing your applications. Thank you so much and have a lovely day.